The overall goal of this procedure is to harvest, concentrate, and preserve low-abundance, low-molecular-weight biomarkers from biological fluids at appropriate concentrations for standard protein assays. This is accomplished by first adding chemically functionalized hydrogel nanoparticles to the biological fluid. Low molecular weight analytes enter the core of the nanoparticle and are captured by different organic chemical dyes, which act as high-affinity protein baits. The second step is to separate the particles from the biofluid by centrifugation. The nanoparticles concentrate the proteins of interest by several orders of magnitude. The nanoparticles are then washed with water to remove extraneous proteins. The final step is to elute the captured proteins from the nanoparticles. Ultimately, Western blotting, mass spectrometry, or immunoassays are used to show the presence and/or concentration of the previously undetectable harvested low-abundance biomarkers.
Generally, individuals new to this method will struggle because the nanoparticles need to be washed and resuspended without clogging the pipette tip. The nanoparticles do not resuspend as easily as a cell pellet, so you must vigorously pipette up and down in order to adequately resuspend the nanoparticles. After the first incubation with nanoparticles, it's important to rinse the walls of the tube in order to collect any nanoparticle residue that may have adhered to the tube.
To perform nanoparticle processing of serum samples, dilute 500 microliters of human serum 1 to 2 with 50 millimolar tris HCl.